Hello guys, very warm welcome to all of you. Welcome to our modern biology. I'm Sasmita. Today we will see a new chapter which is fertilization. So let's get into the video. Before that, please make sure you have subscribed my channel and hit the bell icon for further notification. And if you are new to my channel, then go and watch my previous video. So let's know first the introduction of fertilization. Introduction, like normally human life begins at fertilization. Fertilization, you can simply say that it is a fusion of male and female gamete to form diploid zygote. But the process is not that much simple. This process simply can occur naturally within the body or through the reproductive techniques outside of the body. Let's look the first point. The process of union of male and female gamete by the process of gametogenesis and the fusion of pronuclei or sperm and ovum take place. This diploid zygote form called fertilization. Pronuclei means, like suppose this is a sperm head contain one nucleus, we call it as pronucleus, and the diploid zygote forms. Diploid zygote means, like as we all know, sperm is haploid and egg is also haploid. We denoted it as n number. So when the sperm and egg combines together to form a zygote which is diploid in number, we denoted it as 2n, which is denoted by 2n. Let's look the second point of introduction of fertilization. Neither the sperm nor the egg is active or passive partner. Sperm is activated by the egg and egg is activated by the sperm. And the activation involves calcium ion and membrane fusion. Calcium ion and membrane fusion. And the fertilization in mammals simply occur in oviduct. It is viable for approx 24 hours after ovulation. It is viable for 28 hours after ovulation. The next, site of fertilization. In mammal, fertilization is internal, as we all know. External in case of frog. External fertilization occur in case of frog. Mammals basically take place in ampullary region of fallopian tube. It occurs in ampullary region of fallopian tube. So let's know about the sperm. Before moving into the fertilization, the mechanism of fertilization, we have to know about the sperm and about the egg. So in this slide, we will know about the sperm. A sperm, Leeuwenhoek is a scientist who had discovered the sperm in the year of 1677. 1677, Leeuwenhoek had discovered the sperm. Sperm are produced in the testes. We all know about that. Sperm contain four regions. One is head region, another one is neck, then middle piece and tail region. Sperm head contain a haploid nucleus and an acrosome. I had told this in our previous slide that sperm head contain one haploid nucleus and an acrosome. Acrosome derived from a Golgi apparatus and contain enzyme which is used to break vitelline membrane of egg. Acrosome in the acrosomal reaction take place, it release some enzyme which is used to break down of vitelline membrane of the egg. Middle piece contain mitochondria and centrioles which generate microtubules of the flagellum which help in the flagellum movement. Energy for the flagellum movement comes from the mitochondria ATP. Here you can see a structure of sperm, and the top region is head region. Here is the head region. Here is the neck region. Here is the neck region. And here is the middle piece region, and here is the tail region. In the head region, you will see A plasma membrane which cover the total head region is the plasma membrane, and a acrosome which is present the top of the head region, and behind that you will see nucleus containing chromosomal material, like it is a genetic material which is present in the head region, and in the here is the neck region, and here is the middle piece. You will see a mitochondria which is present in the middle region, which gives energy for the swimming, like for the movement of flagella, like for the movement of tail. You can see in this picture clearly. Here we will know about egg. It is produced from ovary. It is produced from ovary. The female gamete contain haploid nucleus in mammal early stage of zygote, and it has large mass of cytoplasm. It has large mass of cytoplasm and haploid nucleus in it. In mammalian, the ovum is covered by thin vitelline membrane, which is further covered by another primary membrane called zona pelucida. That the female gamete contain four layer, which is the outer layer is known as corona radiata, then zona pelucida, after that previtaline space, then oocyte in it, like it contains some protective agent, because of it it will survive in particular environment. For that reason it has so many protective layer in it. You can say one picture I have inserted of ovum. Here is the corona radiata, which is present very outer side of the like ovum, which protects the cell from any like damage and all. So here is the zona pellucida, which is present in the second layer, and here is the vitelline membrane, which is present beneath the zona pellucida. So here I have inserted one more picture of ovum. You can see, as we know in our previous slide, that corona radiata present in the very outer membrane of the ovum. Here you can say corona radiata. Here you can say corona radiata, which is present very outer membrane of the ovum, and it is also known as follicular cell, which protect the inner layer of the ovum and helps to prevent polyspermy. It also helps to prevent polyspermy. Here is you can see nuclear follicular cell, nucleus of follicular cell. It is present inside that corona radiata, which is present inside that corona radiata. Nucleus of follicular cell, which is present inside that corona radiata. Then, as we know in our previous slide, after corona radiata, we will find zona pellucida or jelly coat. Here you can say, here is the jelly coat, is present beneath of the corona radiata. It contains glycoprotein that protect the inner contain of the ovum until acrosomal reaction of fertilization help to prevent polyspermy. It also prevent polyspermy. It contains glycoprotein that protect the inner content of the ovum, and until the acrosomal reaction of fertilization, it helps to protect the inner content of the cell and prevent polyspermy. Then we will find a cortical granule which is present beneath the zona pellucida. Here you can see cortical granule which is present beneath the zona pellucida. It releases enzyme during the fertilization and harden the zona pellucida and digest binding protein on sperm to prevent polyspermy after a sperm cell has entered the ovum. It will harden, it will release its enzyme and harden the zona pellucida and prevent polyspermy. It will harden that zona pellucida layer. After one sperm will enter the ovum, it will release its enzyme and it will harden the zona pellucida. So that's the reason no any other sperm can penetrate through that zona pelucida. Then we will see mitochondria. As we know, mitochondria is a powerhouse of every cell. It produces ATP. Then we will see cytoplasm. Here is a cytoplasm layer. You can see in the middle portion is a cytoplasm layer of the cell. Then we will see haploid nucleus. Haploid nucleus, why haploid nucleus? Because we discussed this in our previous slide, that every gamete, like every male and female gamete, contain haploid nucleus, which is denoted by N. Like both, when fertilization occur, both male and female haploid gamete combines to form diploid zygote. So it is haploid nucleus. So here in this slide we will see some factor which will going to help the sperm for the fertilization, and some factor which will act as a barrier. So first we will see some helper which will help first we will see some factor which will help the sperm for the fertilization. First one is the lashing movement of the tail. As we know in our previous slide that the middle portion of the sperm contain mitochondria which will help the movement of flagella in the sperm. Suppose this is the head of the sperm and this is the middle portion, and this is the tail region. It is the head region, it is the middle portion which is contain mitochondria, and it is the tail region, or you can say this is the flagella, and the middle portion contain mitochondria which will provide ATP for the movement of tail, for the lasting movement of the tail. So it will help for the fertilization. This sperm or this mitochondria which will give the ATP for the tail movement, it will act as a helper for the fertilization. The second one is prostaglandin. It is a chemical which will help us or act as a helper, which will help the contraction of vaginal part and uterine part, and those contraction is in reverse direction, like from the vagina to the uterus. So that will help the sperm for the movement upward direction. Then the last one is aspiratory movement of uterus. Aspiratory means it's a sort of suction movement. This will help the movement of sperm from the vagina to uterus. So these three will act as a helper for the movement of sperm in the fertilization, for the fertilization. And then the next thing will going to act as a barrier. First one is a thick lining, the thick line, the thick endoplasmic lining. The 14th day of menstrual cycle, the egg is released. Egg is going to remain in that fallopian tube just for 2 days. At that time, the endoplasmic lining is very thick and folded like structure. That means the sperm have to swim through that endoplasmic lining, which act as a physical barrier. And the endoplasmic lining is very thick, which sort of thick and folded like structure like this, but sperm have to cross through that lining. Sperm have to crush through that lining, so it acts as a physical barrier. Another one is acidic pH. Acidic pH, the female reproductive tract, the this pH, acidic pH kills the sperm. Then the third thing is phagocytic activity. This phagocyte in the female reproductive tract which will engulf the sperm. So these factor will help, will act as a helper and will act as a barrier for the fertilization. Now as we know, there's so many there is so many sperm produced during the ejaculation process, 3.5 millions or more than that. only few, like only 100 sperm can able to penetrate through that able to only 100 able to cross through the plasma membrane and reach to the egg. Like if the sperm quality is very low, then no sperm will able to reach the egg. So there will be no fertilization or will occur. So this condition will known as oligospermia. Oligospermia is a condition where the sperm quality is very low and the sperm can't fertilize with the egg, so fertilization can't occur. In this slide, we will see how egg release from the ovary and what will be the next step after egg release from the ovary, and how sperm will fertilize the egg and what will be the further step. Here you can say it is a vagina, and here is a vaginal cortex, and here's the uterus, and here's the ovary. So ovary release or produce egg during the menstrual cycle, and each month when it ovulate, it release egg called oocyte, or you can say it is ovum, and the process through which it release egg is known as ovulation. As we know in our previous slide, oocyte surrounded by a layer which is known as zona pelucida is a thick layer which is surrounded by the egg, and the zona pellucida is also surrounded by a thick layer which is known as corona radiata. Corona radiata, in the blue dot you can see here is a corona radiata. If it is transported through the UT, means uterine tube, UT, here is the UT, and it is also known as follicular tube. If it is transported through the egg, is transported through the uterine tube, uh, through it when it is transported through the uterine tube, sperm meet the egg and the fertilization occurs. Sperm meet the egg and fertilization occur. If penetrate the sperm penetrate through that zona pellucida and for cause fertilization and to form zygote, diploid zygote. Here you can say it is formed diploid zygote, and zygote is fertilized egg, and this occur in day 1. This is occur in day 1. This occur in day 1. Then sperm DNA and oocyte DNA mix together and literally form one DNA. Like in zygote, sperm DNA and egg DNA mix together to form one DNA in a zygote. Then after that what will happen, then it divided like one cell will divided into two cells, then two cell divided into four cell, and four cell then divided into 8 cell. Here you can see, then 8 cell divided into 16 cell. This whole process occur in the fallopian tube. This whole process occur in this fallopian tube. Like the zygote form two cell, one cell divided into two cell divided into four, then 8. This whole process occur in the fallopian tube. We known this process is known as cleavage. We call this process as cleavage. But when it divided when it divided, the structure will not get increased. Only the cell get divided, and one cell divided into two divided into four. But the structure not get become bigger. When this cell develop and reach 16 cell, it called as morula. When it develop into 16 cell, we call it as morula. And this stays in the day 4. This stays occur in day 4. And this morula divide again to form blastula. Here you can see, here's a blastula. Morula divided to form blastula. Blastula occur. Blastula, when the morula divide to form blastocyst or blastula, collect of 32 cell. It is collection of 32 cell. It is formed in the day 5. Day 5. And this blastocyst implanted into the uterine wall. It is implanted into the uterine wall. After implantation, it growth like a zona pelucida like shaded out. It is occur in day 7. The whole process occur in the day 7. Now we will see what are the factor, like what is the step for the fertilization. First, activation of sperm in the female reproductive tract, and binding, sorry, this is the third one, and this is the second one. Chemo attraction of the sperm to the extracellular envelope of the egg, then binding of the sperm extracellular surface of the egg, then the exocytosis of acrosomal vesicle, then passage of the sperm through the extracellular envelope, then fusion of egg and sperm cell membrane. Like as we know in our previous slide that the fertilization occur in the female reproductive tract, where sperm activated and it is chemo attracted towards the egg. Then the next step is the binding of the sperm to the extracellular surface of the egg in the zona pellucida. Then third is the acrosomal reaction, and the fourth step is the not fourth step. Fourth step is acrosomal reaction, and the fifth step is passing of the sperm through the extracellular envelope, and the sixth step is the fusion of egg and sperm cell membrane. Then fertilization will occur. So now in this slide we will see zona pellucida. Zona pellucida. When sperm activate, as I had told you in our previous slide that sperm activate in the reproductive tract. When sperm activate in the reproductive tract, it will going to bind zona pellucida. Then acrosomal reaction take place. And zona pellucida has two function, which is bind of the sperm and then acrosomal reaction. It will cause acrosomal reaction. Then zona pellucida made with 3 glycoprotein, which is ZP1, it is ZP1, ZP2 and ZP3. Specifically ZP3 bind with the sperm and it will initiate acrosomal reaction. ZP3 bind with the sperm and it will initiate acrosomal reaction. Then if you will ask me how sperm will get bind with zona pellucida, first interaction occur between the sperm protein and component of zona pellucida. First interaction occur sperm protein and zona pellucida component. Then sperm surface protein, which is which is sperm surface protein, SAID1 bind to the zona pellucida complex. It will going to bind with zona pellucida complex first. Then sperm girt cross-link tightly and specifically N-acetyl glucosamine residue on ZP3. Girl protein, which is present in the top, head region of the sperm, it will cross-link with the N-acetyl glucosamine residue on ZP3. As I had said in as I had said in earlier that ZP3 will bind with the sperm. So ZP3 contain N-acetyl glucosamine. So GALT protein, which will cross link with N-acetyl glucosamine residue, which is present in the ZP3 region. So here is the overview of zona pellucida. Hope you will clear. Hope you are clear now. Then we will go to our next slide. Let's understand fertilization diagrammatically first. One ejaculation, about 3.35 millions of sperm are deposited and released into the vagina. As there are so many sperm released in the vagina, only one sperm will fertilize with the egg. Only one sperm will fertilize with the egg, and that sperm will face so many barriers for that to reach the vagina, to reach the egg. what will happen exactly? One sperm will get contact with the egg first. One sperm will get contact with the egg. After get contact with the egg, like acrosome, which is present in the head region, acrosome which is present in the head region, it will release its enzyme called as lysine. It will release its enzyme. uh, Acrosomal enzyme, it will digest zona pelucida. The acrosomal enzyme, it will digest zona pelucida. It will cause acrosomal reaction. Clustering of girt protein in the sperm cell membrane activate G protein, activate G protein, which open calcium ion and initiate acrosomal reaction. After that, what will happen? Acrosomal enzyme, it will digest GDP. Then what will happen? Sperm plasma membrane and egg plasma membrane will get fused with each other. Sperm plasma membrane and egg plasma membrane will get fusion with each other. After fusion occur, what will happen? Then the cortical granule, which present inside of the egg, which you can see over here, cortical granule, which is present in inside of the egg, they fuse with each other. Cortical reaction take place. They fuse with each other and release outside, like release vitaline, the like space. After release it in the vitaline space, it will harden the zona pellucida to prevent polyspermy. This is the whole process of fertilization occur. So guys, this was it for today's video. I have only covered fertilization part. I haven't covered my cell biology part. I will do my next video on cell biology. So keep watching. If you haven't watched my previous video, then go and check that out. And thank you so much for your love, guys.